Hello everyone, welcome to the channel Microbial World. Today we will be discussing about bacterial growth curve. When we provide the right conditions like food, correct temperature, these microbes they can grow very quickly. And it is very important to have a knowledge about their growth so that we can predict or control their growth under particular conditions. So in higher organisms, growth means it is the increase in the size and volume of organism. But in case of bacteria, growth means it is the increase in the number, that is the increase in the number of bacteria. Now, a fresh liquid medium it is taken and we inoculate it with a given number of bacteria and then incubate it for sufficient period of time. So it will give us a characteristic growth pattern of the bacteria. We can measure the bacterial population periodically and then plot the log of number of viable bacteria against time in a graph. And this will give us a characteristic growth curve which is known as growth curve or growth cycle. Bacterial division. So the bacteria it divides by the process called binary fission. Okay, so in binary fission, a single cell, it will split into two equally sized cells. So this process, it begins with cell elongation. The cell will first elongate. This includes the enlargement of the cell membrane and the cell wall. The cell volume will also increase. And then the cell, it will replicate its DNA to prepare two copies of its chromosomes, one for each newly formed cell. The protein FTSZ, it is essential for the formation of septum. And the septum, it is initially seen as a ring in the middle of the elongated cell. The nucleoids will then be segregated to each end of that elongated cell. The septum formation, it will be completed and this will divide that elongated cell into two equally sized daughter cells. And this entire process, it will take as little as 20 minutes for an active culture of E. coli bacteria. Growth curve. So in a closed system or batch culture means no food is added, that is media is not added and waste is also not removed. So in this closed system or batch culture, the bacteria, it will grow in a predictable pattern and that will result in a growth curve. So this growth curve, it is composed of four distinct phases of growth. That it includes lag phase, then there is log phase which is also called exponential phase, then there is stationary phase and death phase which is also called decline phase. So this growth curve, it will yield generation time for a particular organism. So what is generation time? Generation time, it is the amount of time it takes for the population to double. That means one cell, it becomes two cells, then it becomes four cells, 8 cells and so on. So it is the amount of time it takes for that population to double. Then the details that is associated with each growth curve, that is the number of cells, length of each phase, rapidness of growth or death, overall amount of time, this will vary from organism to organism or even with different conditions for the same organism. But the pattern of the four distinct phases of growth, it will remain same. Okay. This is the growth curve. So you can see here the log of number of microorganisms, it is plotted against time. So first you can see there is the lag phase, then the exponential phase or the log phase, then comes the stationary phase and finally the death phase or the decline phase. So now let's discuss each of the phase in detail. Lag phase. So when the bacteria it is inoculated into a fresh media, the bacteria does not divide immediately. It will take some time for the bacteria to adjust to the new environment. So this lag phase, it is an adaptation period for the bacteria. Okay, so this time period where the bacteria is metabolically active but it does not divide is called lag phase. And also during the lag phase there will be no increase in the number of the cell. But the size of the bacteria it will increase continuously and the bacteria will have the largest size at the end of the lag phase. And we also said the microorganism, it is trying to adapt to the new environment during the lag phase, right? So what they do during this time, they will synthesize enzymes, coenzymes, RNA, and all the essential metabolites that is needed for physiological activities like growth factors, macromolecules and all. And they are also trying to adjust to the environmental changes like change in temperature, pH, oxygen availability. Okay, and also the duration of the lag phase, it will vary according to the conditions and species of bacteria. Now if the culture organism, it is taken from an old culture, then the duration of the lag phase will be longer. Whereas if the culture is fresh, then it will have a short lag phase. And also if the culture media is different from the previous culture, then the duration will be long because it will take more time for the bacteria to adjust to the new media. And at the end of the lag phase, the bacteria will be fully prepared for cell division. Log phase or exponential phase. So during this log phase, the bacteria, it will divide continuously at a constant rate, and the number of bacteria will increase exponentially. And also all the bacteria, it will be in a rapid stage of cell division and they will show balanced growth. So due to this rapid cell division, the bacteria will have smallest size during the log phase. And the bacterial population, it will be uniform in terms of their metabolic activities, chemical composition of cell and other physiological characteristics. And also the biochemical and physiological characteristics that is used for the identification of bacteria can be manifested during the log phase of growth. And the generation time of bacteria is also determined during log phase. We have said earlier about generation time, right? That is the amount of time that it will take for the population to double. So this generation time, it will not be same for all bacteria in the culture, okay? And the generation time, it is shortest during the log phase and it is dependent upon growth factors that is present in the medium. And the log phase, it will last for several hours, and how much time the log phase will last, that will depend upon the type of organism, conditions of growth and density of organism. Stationary phase. So at some point the bacteria, it will run out of an essential nutrient or chemical, or the growth of the bacteria, it will be inhibited by its own waste products or due to lack of physical space. So what happens? The cells, it will enter stationary phase. So we can say stationary phase as a state during which there is no net increase in the bacterial population. Okay, a constant bacterial population will be maintained. So there will be a balance between cell division and cell death. That means the number of new cells being produced will be equal to the number of cells dying off. And in case of some bacteria, complete cessation of cell division will occur. The growth will completely stop. So there is no net increase or decrease in the number of bacteria. And why does stationary phase occur? It occurs when the nutrients are depleted in the medium, or when toxic secondary metabolic waste accumulate, or due to increased bacterial cell density. And certain bacteria, they produce antibiotics such as penicillin, streptomycin and enzymes during the stationary phase. And in case of endospore forming bacteria, sporulation will occur when the bacteria enter stationary phase. Death phase, which is also called decline phase. So during the death phase, the number of bacteria will decrease continuously and exponentially. The total count of the bacteria may remain constant, but the viable count will decrease. So what are viable bacteria? Viable bacteria means they are live active bacteria that is capable of growing, reproducing and carrying out metabolic processes. So during the death phase, the viable count will decrease. Okay. And this death phase, it is just the inverse of log phase. But the death rate, it is slower than the growth rate. Okay. And why does death phase occur? It occurs because of depletion of nutrients and accumulation of toxic waste. And another thing is that all the bacteria do not die at the same rate. Some bacteria will die faster, some bacteria it will be more resistant and they will remain viable for longer time. An example is spore forming bacteria. Okay, so we have discussed the 4 phases of growth, that is the lag phase, log phase, stationary phase and death phase. This is all about bacterial growth curve. I hope that the topic was clear for everyone. Do comment your doubts and subscribe the channel for more informative videos. Thank you.